Hello, Toll students. I hope you guys are doing well today. This is Mr. Garcia, and I'm going to go over protein synthesis basics. This is just a basics video. I'm not going to go into too much detail on this one. So what is protein synthesis? Protein synthesis is the creation of proteins in your body. Examples of protein in your body are hemoglobin. This carries the oxygen in your blood. Keratin in your skin, nails, and hair. Or collagen that makes up your bones, tendons, and ligaments. The building blocks, or the monomers, of proteins are amino acids. A chain of amino acids makes one protein. Protein synthesis involves two major steps, transcription and translation. So if you look down here where it says transcription, just look at trans and then C, trans and then L. Remember in the alphabet, C comes before L. So transcription comes before translation. C comes before L. RNA is a nucleic acid like DNA. RNA has uracil instead of thymine, so this time it's U instead of T. A will pair with U. C and G are still the same. RNA can leave the nucleus, and DNA is unable to. Remember this, that RNA can leave the nucleus, DNA is too big, so it cannot leave the nucleus. And the name of the sugar in RNA is ribose. Remember in DNA, it's deoxyribose. In RNA, it's ribose. You need to know these three types of RNA. mRNA, which stands for messenger. So if you look over here, this mRNA carries a message from DNA. That's why it's called a messenger. It has the message from DNA. tRNA stands for transport. The T is for transport. Just look over here at this really messed up T. That's how I remembered it. It's a really messed up T. But you have here the straight line and then the cross looking thing right here. T. Okay, this is called a transport RNA because it transports amino acids. This little ball on top of it, each one is an amino acid. rRNA stands for ribosome. If you look over here on this blue thing, this is a ribosome. They make proteins. About 70% of this is made out of rRNA. Okay, so codon and anticodon. So codon and anticodon. Every three bases, so every three here over here in mRNA, the three bases in mRNA is called a codon. 1, 2, 3, codon. 1, 2, 3, codon. 1, 2, 3, codon. Every three bases in tRNA, over here, remember the messed up T, tRNA, 1, 2, 3, is an anticodon. The codon will always pair with an anticodon. And they have to be matching. So G goes with C, A goes with U. Remember, we are using RNA, not DNA. So A goes with U. C and G always still go together. DNA or RNA, C and G will always go together. Again, the two major steps in protein synthesis are transcription, translation. The letter C comes before the letter L, so transcription is first. Transcription. Transcription is when DNA transcribes the message to mRNA. Okay, so here we have DNA. DNA will give the message, the genetic message, so TAC. T is going to give it to mRNA, but instead remember, T is going to get changed to an A gets changed to U, C goes with G. T gets changed to A's get changed to U's. So again, one more time, A's get changed to U's, T's get changed to A's. DNA gives the message to mRNA now. Now, one thing to remember is that DNA is still there. It doesn't get destroyed. It just creates a copy for mRNA to carry. Once it has the message, mRNA will leave the nucleus to go to the ribosomes. Translation. The next major step is translation. This is where mRNA goes to the ribosome. So this green thing right here is a ribosome. mRNA brings the genetic message from DNA into here, and it's going to bind with tRNA. So remember, every three is a codon. We have A. this codon has to match with the anticodon in tRNA. So A goes with U, A goes with U, A goes with U, G goes with C, so on and so on. They have to match, and each one carries an amino acid. Once we do this enough, we're going to get a chain of amino acids. Okay, and this chain of amino acids is going to create a protein. So again, every 3 pairs is a codon in mRNA, every 3 in tRNA is an anticodon. They have to match with the correct matching pair, and it's going to create a chain of amino acids up here. Okay, for a complete picture of protein synthesis, we start over here with DNA inside the nucleus. So this purple thing is the nucleus. We're in the DNA. DNA gives the message to mRNA. mRNA leaves the nucleus into the ribosome, and here translation takes place. Translation is when tRNA brings anticodons and amino acids over here and it matches with the mRNA codon. Codon matches with a corresponding anticodon, and then the amino acid is bonded together with another amino acid to create a protein. A chain of amino acids is a protein. And then a different picture, same thing, different picture. DNA gives the message to mRNA. This is called transcription. mRNA goes to the ribosomes and we create proteins. This is called translation. If you guys have any questions, feel free to email me.